Hello, welcome to the Trailblazer Research Seminar Series, a production of the NUS MedSci Alliance. This video was recorded on the 11th of February, 2026 at NUS, with our speaker, Professor Lou Boxiarng, sharing his research journey about identification of disease target genes in the post-GWAS era. Let's tune in and see what he has to say.
Hi everyone, my name is Bo-Xiang Liu. I'm a presidential young professor at NUS and principal research scientist at A-STAR. Today I'll be talking about identifying target genes in the post-GWAS era. Thank you for coming.
So good afternoon, everyone. I'm Catherine Ong, Assistant Dean for Research at the Yong Lin School of Medicine. It's my pleasure today to introduce our PYP Assistant Professor, Liu Bo-Xiang, to kickstart the first series of our Rising Stars. And this is a series that's organized by Prof. Tai from the MedScience Alliance Council. And so Bo-Xiang is PYP, or a Presidential Young Professor over in NUS. He's currently from Department of Pharmacy and Biomedical Informatics. And, Bo-Xiang is, has been authored, senior author in a lot of high-impact publications like Nature Genomics, Nature Communications, and also had been awarded the NUHS Young Investigator Award, Young Scientist Award. So a lot of, accolades to his track record. And so today we are very honored to hear about his research on GWAS, post-GWAS related. So the floor is yours, Bo-Xiang.
Mic check. Okay, thank you. So first of all, thank you so much, Catherine, and for this very nice introduction. And also thank you for Prof. Tai for inviting me to give this talk. So I guess Catherine talked about some of the accolades that I received, but I want to say that all these are team efforts. So I've received a lot of mentorship from senior PIs, and also I've given mentorship to my team, which ensures their productivity. So this is truly a teamwork. So before I get started today, they asked me to share my personal journey, which I will do, but I'm seeing a lot of senior faces in this room as well, so I'm not sure if you're interested in learning my personal journey. It won't help you. So instead, I thought I'd share my research first and foremost. I think this will be interesting to the majority of you, and I have one slide to share sort of my personal journey. I hope that's okay with you all. So I'm trained as a geneticist. I've sort of ventured into single-cell and functional genomics later in my career. So today I will be talking about identification of disease target genes in the post-GWAS era. So when we started doing research circa 2010, this is a number of genome-wide association loci that was in existence, right? So you see that there's loci here and there. You see there's sparse association signals. So just to put this in numbers, in 2007, there's about 24 top associations, 1 full summary statistics, and 1 publications. And this is what we have in 2025, right? So there's more than 1 million top associations, more than 140,000 full summary statistics, and about 7,000 publications, right? So the field has seen an enormous shift from the lack of association right here to too much association in 2025. So we have seen a shift in priority in human genetics. In 2007, our priority is to identify more GWAS loci, to identify more targets. Whereas in 2025, there's too many loci, and our target has shifted to identify more target genes instead of these associated loci. So having trained as a biostatistician and bioinformatician, I like to position ourselves in the lens of 2 key parameters, right? So the first parameter is precision, and the second parameter is recall. And I assume most of you are familiar with these 2 metrics. And I'm going to make a controversial argument that we're actually not doing so well in either axes. And I'll tell you why in a second. So first of all, let me give you a brief overview of what has been done in the last 10 years. So there has been a very influential project called the Genotype Tissue Expression Project, or GTEx for short, which sampled hundreds of individuals across all the major human tissues. So this is an enormous multinational project that spanned almost all the continents and hundreds of institutions. And this concluded in a number of highly influential publications, one of which is this GTEx 2020 on Science. So one of the main conclusions from that paper is this figure, right? And I'm going to explain this figure in depth. The x-axis is the negative log 10 p-value. So the higher the x-axis, the more significant the association. And the y-axis is the proportion of SNPs that are associated with at least one gene, the expression of at least one gene, right? So let's look at a few important points. This point, if you look at the x-axis, is 0.05, right? So this is the normal nominal cutoff that people use. If we use this nominal cutoff, then 97% of SNPs will be associated with p-value of less than 0.05, which means that almost all the genomes across millions of variants, most of them will be associated with the expression of at least one gene. So of course you ask, no one would ever use a p-value cutoff of 0.05, and you'd be correct. So people will be using this multiple hypothesis corrected p-value, but still 64% of SNPs will be associated with the expression of at least one gene. So what this tells us is that once we have already reached a stage where there's just so much data and so much association that if we look at any SNP, more than half of the chance this SNP will be associated with the expression of at least one gene, right? So this means that we're not doing great. We're doing great on recall, but we're not doing great on precision, right? Because we do not expect more than half of the SNPs to be associated with the expression of at least one gene. And we're not doing great on recall either, right? So most genome-wide association loci don't have a validated target. So I'm going to show you a bird's eye view of this argument. 43% of GWAS loci colocalize with GTEx QTLs, which means that 60% of them don't, right? So if you see this figure, there are some diseases that have quite good colocalization, but those diseases are not common, right? So more commonly, what you see is that for most of the diseases, the association, the power to be explained by QTLs is actually quite low. For some diseases like lupus, only 20% of the loci can be explained by QTLs. So just to drive this point home, because a lot of you in the audience studies cardiovascular diseases is a key area for Singapore, there has been about 600 cardiovascular disease loci identified to date, but only 27 or 4.5% has validated disease genes, okay? And I have listed all of them here. So basically for the majority of the GWAS loci, we still don't have an answer. So in the past number of years, both as a PhD student and an independent PI, we have learned a few lessons of how to move us from this point gradually upward and gradually to the right side, eventually to the top right, right? And I'm going to share you a few lessons. I might cut short on some of these lessons because of the time constraint, but let's start from lesson 1. So novel QTLs are often cell type specific, right? So I'd like to highlight this study which we conducted in collaboration with Ken Hom and Sham and also many other talented investigators. So here what we did is that we collected a cohort of 503 individuals of broad Asian ancestry, including East Asian, Southeast Asian, and South Asian. And we have sampled 1 million single cells across many cell types. So here is the list of the cell types, and I'm not going to detail because of time constraints. And what we can do is that we can couple their transcriptomic profile with their gene expression, And we can map these things called QTL, as I mentioned before, which is essentially how genetic variants influence their gene expression. So I'm going to highlight a few things here, right? So I think it's best to tell stories. So I'm going to give you one story here, which is an association signal that we would have missed if we did not have cell type specificity. So here what we have discovered is that there's two independent variants acting in two separate cell types, closely related but distinct. Okay, so these two cell types are a naive CD4 T cells and also memory CD4 T cells. So you see that these two variants are distinct, but they're regulating the same gene. Okay, so how can we be sure they're independent? I'll show you two evidence. The first one is that these two lead SNPs are 36 kb apart. And it's highly unlikely that an enhancer can span this length. So it's very likely that they're two independent enhancers or other regulatory elements. And the second thing is that if we look at the LD of these two SNPs, they're actually quite low. Their R squared is less than 0.3, right? Which means that it's unlikely for a SNP to hitchhike on the other SNP. So then we did a further digging, right? So we identified these two SNPs, and then interestingly, we found that these two SNPs not only regulates the expression of this gene, which I'll talk about later, but also regulates the splicing of a different gene. And what's more interesting is that the second gene is on a different chromosome. All right, so here, I'm not sure if it's clear from the figures, but this is on the same chromosome. You see that they're on the same line. But these dotted lines and this gene is on a different chromosome, right? So these two exact same SNPs not only regulates the expression of this gene, but also a splicing of another gene on a different chromosome. I won't go into the details about these technical results. So we did further digging, right? What eventually happens is that this gene is called HNRMP-LL, and this gene is actually a RNA binding protein. So when its expression is regulated by these two SNPs, its protein production is going to be regulated upwards or downwards as well. So when this protein gets produced, it's going to come in and bind to exon 4, 5, and 6 of this gene on a different chromosome. And it's going to mark it for inclusion or exclusion. And we can validate this result because we have single-cell data, we can validate this result by stratifying the cells of whether they have expressed HNRMP-LL or did not. And you see that there's a significant difference between these two cell types in terms of their splicing. Okay? So, but that's not the end of the story, because some of you might recognize this gene. This gene is called PTPRC, and I've put in parentheses here, there's another name for this gene called CD45. So for those of you who study rheumatology or immunology, you know CD45 is a key marker for T-cell differentiation. So we naturally ask the next question, do these two exact same variants also influence T-cell proportions? And the answer is yes. We see a significant correlation between these two variants and cell type proportions. So essentially, we have mapped out this simple but complex but complete pathway using just single-cell data, right? So these two variants would influence HNRMP-LL, which would come in and bind CD45 exon 4, 5, and 6, which will eventually influence T-cell proportions. But notice that this is not possible if we did not have single-cell data, because these two cell types would be merged into the same data file. So it's only possible to do this with cell type resolution. And the second story, which is an extension of the first story, is that not only do we have to look deeply into the cell type, but we also have to look deeply into the individual ancestries. So being in Asia, I think we're sort of privileged, especially in Singapore, we're privileged because there's multiple ancestry in the same country. And I guess we're privileged and unprivileged at the same time because Asian ancestry in itself is generally considered underrepresented in genetic studies. 80% of genetic studies focus on one population, which is the European population. And there's a good reason for that, because Europe and North America are well funded to do this genetic research, whereas Asia is catching up, but catching up very quickly. So, and Singapore has a distinct advantage because it collects about 94% of all the genetic diversity across Asia. So it's a really ideal place to study Asian genetic diversity. So here I'm going to show you what's really made possible by studying genetics in Singapore. And we would not have seen this insight if we were only studying European populations. So here I'm going to explain this figure first. Using our pipeline, we're able to identify the disease genes, their relevant cell types, and the disease that they're influencing. So let's take one example of this gene called TCHP. So there's a variant that influences TCHP splicing, and this variant would act through these 1, 2, 3, 4, 5, 6 cell types, for example, CD4 T cells and natural killer cells, to influence Graves' disease status. Okay, so, and you see that circling back to the cell type specific argument, that we do see quite significant cell type specific signals. So here, this is Graves' disease GWAS. It's colocalizing with monocytes, natural killer cells, CD4 T cells. And then when you get to naive B cells, then this signal disappears. To summarize these findings, we have made this plot. You see that among the 22 cell types, only the top 7 has this disease signal. So if we had looked at bulk tissue data, then we wouldn't be able to find this signal. But what's more interesting is that this variant is an Asian-specific variant. Okay? So I'm showing you the gene plots here. So this is the structure of the TCHP gene. It's a multi-exonic gene. And here is the exon 4 of the TCHP gene. And the variant here, RS744, is sitting on top of the splice site. But this variant only exists in East Asian populations. So here you see that the population frequency is high in Chinese, Japanese, and Korean, and it decreases in Malays and will be absent in Indians. And we can replicate this in 1,000 genomes where we see a sufficient allele frequency in East Asians, but it's absent in the other 4 major populations, including Europeans, Africans, Native Americans, and South Asians. So we validated this variant in collaboration with Donald and Xavier. What they did was that they cloned out this TCHP exon 4 and its surrounding 200 BP introns. And then they made 2 identical copies of this mini gene, differing only at one site, which is this RS744. So you see that if we have a healthy or non-risk allele, it's going to produce us the regular healthy isoform, whereas if we use the risk allele, it's going to lead to intron retention, right, which leads to nonsense-mediated decay. But this is not possible if we're not looking at the Asian population, because this variant would have been missed if we only look at European populations. So lesson number 3 is disease-relevant treatment will often offer new insights, right? So I'd like to sort of give you some background in the disease that we study. So here we're studying obesity and diabetes. And you know that obesity and diabetes is an interplay between multiple factors, right? Genetics plays about half of the role. 40 to 60% of disease risk is heritable for obesity and diabetes. And also lifestyle plays an important factor as well, right? How much McDonald's you eat, do you exercise regularly? And also here I'm distinguishing between the lifestyle, which is things that we can change, versus the environment, which is things we cannot change. For example, floods, acid rains, pollution. But in Singapore, we're quite privileged, right? Because the air is clean and the rain is well controlled. So here we're interested in genetics and lifestyle interaction. And before I talk about our research, I'd like to highlight some previous research, and just to give you an idea of how difficult it is to do this type of study, right? So here in 2024, you have Gilead's group have published this paper on cell genomics. What they did was that they took 99 captive baboons, and then they raised them for a number of years, right? But this study lasted 2 years. So what they did is that in the beginning, they feed these baboons low-fat diet, so regular diet. And then for 2 years, they feed these baboons with high-fat diet, right? So high-fat, high-cholesterol diet. And then did a comparison before and after the study. So before the study, they took their blood and multiple tissues. And after the 2 years, they took their blood and the same set of tissues. And they did genotyping and RNA sequencing, right? And then they found really interesting genetic regulatory patterns that are different before and after this treatment. But just imagine how difficult it is to babysit 99 baboons for 2 years. Okay, it's very, very challenging. It's a very, very challenging study. You need a large yard, right? Almost you have to open a zoo to do this. But that's this is the best study that's to date. People have done this in mouse, in rats, in bamboos. No one has done this in humans, for the good reason, because humans are the worst, being lab animals, because humans are not compliant, right? For example, we can, even if we had a zoo, we cannot put the humans in that zoo, right? So here we took this challenge head on. And the people who really did the hard work is Yishong and Xueling and Wenjing. So what we did is that we started with recruiting obese people, and we give them a motivation, right? Because they're obese, then they're naturally motivated to lose weight, we hoped. Right, so we give them an incentive, which is that we give them a free 16-week weight loss program with exercise. We pair them with an exercise coach. So for each week, they're going to go into the gym for 3 sessions. Each session is 90 minutes. And then we also do food recalls. Okay, so, and then before and after the 16 weeks, we collected their blood and their skeletal muscle, and also the same thing after. So just to sort of give you an idea of how difficult this is, right? We started with 265 and ended with 54. So most of the people escaped our cage. Okay, so that's really why people don't want to do this, because it's really challenging, and you get very small numbers even if you start with a large number. But the result for those people who sticked around is actually really, really good. So you see that this is the weight loss before and after the exercise program, and it's only 16 weeks. Okay, so before and after, there's about a 10% on weight loss on average, which is almost like taking semaglutide, right? So the target weight loss for taking semaglutide is 15%, and not everyone achieves that. So here, only after 16 weeks of structured exercise and diet programs, people have lost about 10% weight on average. And their insulin sensitivities have gone significantly up. And you see that their body measurement have improved significantly as well, right? So in the blue dots are their lean muscle distribution, and they have unanimously gone up. And then the red dots are in the fat percentage. I like to emphasize one thing, is for people who are fat, they're at a very high risk of getting fatty liver disease, especially chronic fat. These people on average have lost about 60% of their liver fat, right? So they're going to live significantly healthier lives. So besides this patient improvement, we also found some interesting scientific signals as well. Remember I mentioned that it's quite important to look at the conditions. And here we did see that the QTLs before and after the exercise is significantly different. So there's about 621 shared QTLs. and there's about 596, which is almost the same as the number of shared QTLs, but it's only specific to the precondition, and 484 eQTLs that are specific to the post-condition. Okay. And same thing for SQTL. So we identified 263 shared, but we identified more QTLs that are specific to the precondition and the post-condition, right? 286 and 86. So one thing I want to highlight is that we identified these one signal that we think is actually actionable in guiding future treatment plans. And this is a variant called RS181. It is a stop-gain variant for this gene, actinin-3. So what we have identified is that people with 2 copies of the TT allele would be at a better outcome than people who have CC allele or CT alleles, right? So T allele leads to a better outcome, whereas C alleles lead to a worse outcome. And so what happens is that actinin-3 gene is found mostly in elite athletes. And these athletes are better positioned, so they would have fast twitch muscle fibers, and they're better positioned for these explosive sports. But explosive sports is unfortunately not very good for weight loss because you get tired very often, right? What's better for weight loss is you maintain your zone 2 training for over an hour. And in that process, you lose weight. So if you have a lot of, so this is counterintuitive, right? So if you have a lot of fast twitch fibers, you actually are at worse outcome for weight loss because you get tired so quickly because your muscle is exerting so much strength in a very short period of time. So but instead, if you have more slow twitch muscle fibers, you can keep going on longer, and you actually shed more calories, right? So what we think could help the clinicians in the future is that they could actually test whether there is this mutation, or even better, test their actinin basal expression level. And if these people have more fast twitch fibers, then it's probably better to give them HIIT exercises. Have them rest in between, right? Or if they have more slow twitch fibers, then you can give them longer exercise programs, have them run longer, or have them on a spinning on a cycle longer, right? So I'm going to go through this quickly because I think I have maybe 5 minutes left, but I'm going to just very quickly tell you, right? So these 3 facets that I told you are on increasing recall. But we can also, it's very important to also increase precision as well. So one thing that we are interested in, as is the theme of this talk, is to how to identify GWAS target genes using eQTLs. And we're not alone in that journey. There's lots of other groups around the world doing the same thing, right? So here's a summary of all the gene implication methods that was published from 2010 up until now. There's about 20 methods published. And all these methods fall into 3 camps, okay? I won't go into technical details, but I just mentioned the names. The first camp is called colocalization. Second camp is called transcriptome-wide association studies. And the third camp is called Mendelian randomization. But unfortunately, when we run these methods in our hands, we observe significant differences across these 2 methods, right? So this eCaviar is in the camp of colocalization, and this SMR is in the camp of Mendelian randomization. So you see that when we run these 2 methods, they would identify some signals. But across all the signals they identified, there's only one that's overlapping, which is FES. Okay, I'll skip the details about this disease, but I'll just emphasize the point, which is most of their discoveries are not overlapping. And this is concerning to us, right? Because if you have 2 experts telling you different results because their assumptions are different, then as the investigator, you're really concerned, right? Because you do not know which one to trust. And so to make things worse, right? So these two genes that I've highlighted has been independently validated by third-party labs after the publication of this paper, right? So this paper was actually my first PhD paper published in 2008. And then we found this discrepancy between these two methods, and then people got interested in these genes. And then they went off to design mouse models for these genes. So this paper has been validated in a mouse model. You see, SMAD3 regulates smooth muscle cell fate in a mouse model. Same thing as this gene, TCF21. Okay, so people went off and did a mouse study and knocking out this gene, and they found significant atherosclerotic plaque buildup if you knock out this gene. So now we're really concerned, right? Because let's say if we only run eCaviar, we would have missed this signal. If we had run SMR only, then we would have missed this signal. And who knows, are we missing more FAS, CPO1, and FGFR alpha? So I know FAS has been, there's a paper by Prof. Yashu, maybe he's not here, but his group is investing in FAS, and he's gotten very strong results for FAS, right? So we've been missing some of the signals if you only run one of these two methods. And so then this was our starting point. And then we went and digged further, and we found that these methods, their correlation is really bad. Okay, so you see that these colocalization methods, their correspondence to each other is good. Well, not so good, but at least above 0.5, right? So, but there is this 0.51, which got us quite worried. And the GWAS and Mendelian randomization methods, they correspond to each other very well. But the correspondence between colocalization and GWAS is really, really bad. They're all lower than 0.5, right? So now we're faced with. So now this is a bad thing, but also an opportunity, because let's say if we have these base learners, right? So for those of you who study machine learning, you know that there's things called random forest and there's things called boosting. So the basic idea for these methods is that you have these weak base learners, which is trees, decision trees. But if they don't agree with each other, that's a bad thing, but also a good thing. If they do not agree with each other, you can combine these base learners to make a very strong learner. Okay, so you see that we have these 6 base learners, but their results are discrepant. They're disagreeing with each other. So the idea is, can we leverage this to make a better strong learner? And in our case, we can do this in one of 2 ways. The first way is that we can actually increase the discovery power by merging the results. Or, in the case that I showed previously, right, in this case. Or, if we have too many associations, as of today, because there's more than 1 million associations has been published, what we can do is we can take the intersection of these methods, and they will give you significantly better precision, right? So if you merge all these 6 methods, you see that we can get a precision of almost 1, right? So which means you have less things to worry about, you have less things to validate, but every single target that you validate stands a higher chance of being successful. Okay, so essentially that's what we did, and I'm going to skip the window part because this one is too technical, but I'm going to just show you that we have made this software called Locus Compare 2, and what it does is basically to aggregate all these methods and provide you with a unified interface so that you don't have to go and run these independent methods. And it's going to aggregate these results according to your goals. Okay, so if you want higher precision, it's going to do the intersection. If you want higher recalls, it's going to do the union, right? So really we think that there's no one-size-fits-all methods. The investigator should decide what is best for the study. So we're still not here yet, right? But here's where we want to be. And I think the future is really bright and very exciting. I'd like to thank, so these are the papers that I talked about today, and really these talented people is truly teamwork, right? These are the students who have contributed to these studies, and these are our wonderful collaborators. And also I'd like to thank our funding agencies. Thank you very much. Oh yeah.
Okay, thanks so much, Bo Xiang, for the wonderful talk. Floor is open now for questions. Is there anyone who wants to go up to the mic? Or Woon Tian or Shalin, is there mics around? Okay. Middle.
Hello. So you showed that MR-based methods produce some list of potential targets, and colocalization methods produce some list of targets. Are there any, can we kind of hypothesize any biological difference between those lists? So for example, targets found by one methods have some quality that targets discovered by other methods don't have? Or are they just lists we need to merge and intersect?
Yeah.
So that's the question.
Yeah. That's a fantastic question. So the question is, are there any biological differences across these two lists as identified by Mendelian randomization and colocalization? So what we have discovered is that these two methods have different statistical assumptions. So what colocalization does is finding the overlap of causal variants. So in one sentence, I'm going to explain what colocalization does, right? So what it does is that it's going to find map the variant for GWAS. It's going to find map the variants for QTLs. And it's going to multiply these two probabilities together. So let's say if you have a variant that has a high probability for being causal in both GWAS and QTLs, then now after you multiply it, it's going to give you a high probability. That's essentially what colocalization does. This is very different from what Mendelian randomization does. Because what Mendelian randomization does is that it assumes that there's causality from the exposure to the outcome, which is the QTL and the GWAS. So the statistical assumption is quite different, which leads to the difference in their results, right? So because sometimes, just by statistical fluctuation, you'll see there's distribution differences in the GWAS p-value shapes and the QTL p-value shapes. And this sometimes fits better with the colocalization and sometimes fits better with the Mendelian randomization. So it's really, and if you run this QTL again and just leave one out, you get a different distribution, and maybe then it will fit the other model better, right? So that's a very long answer to your short question. What we have discovered is that there's essentially no biological difference, but it's statistical differences that fits the assumption of one model better or the other model better, which is really disappointing to us, right? Because they can all be good biology, but just because we have sampled different individuals, the randomness in the sampling will give you different results. So that's why I think it's much better if we aggregate these methods to give us either more comprehensive coverage or to give us more precision. Yep.
Does that answer your question?
Yeah, hi. Thank you. Very informative talk. So my question, I have two questions. One is, in last month in January, the Google DeepMind group has published their very revolutionary tool called Alpha Genome. So if you're aware of this, from an expert of genome scientist, can you make a comment on this? And the second question is a general one. So in general, what does the integration of AI tools will affect the future GWAS study in your point of view? Yeah.
Yeah, okay. Thank you for that question. So first of all, I like to say that the publication of Alpha Genome is absolutely amazing, okay? it won't be as amazing as AlphaFold, but it's an incredibly good method, and it's truly a technical tour de force. But I also like to mention a few caveats. Okay, so if you look at so without going into the technical details, the improvement of AlphaFold against delta splice, which is not a foundational model, is about 0.7%. Okay, so I think there's a few things that we have to think about, right? We have these well-performing small models, and we have these better performing big models. But the performance improvement from these small models to the big models, for some tasks is great. For the tasks that we're interested in for splicing, is 0.7%, which is very low. So I think to answer your question, I think this is a case by case. For certain cases, for example, AlphaFold, that's truly revolutionary, right? Like nothing else even comes close, right? And there's a very good reason why these two people won Nobel Prize only like 3 years after publishing the paper. For some other tasks, like genome annotation, there's a long way to go, right? We still, for example, if you give me a variant and ask me whether this variant would influence certain regulatory elements or overlap certain regulatory elements to influence splicing, chromatin accessibility, expression, I can give you an answer. But this answer will not be as revolutionary as AlphaFold. It won't be the gold standard answer. The gold standard is still to do an experiment. Okay. Yep.
Any other questions? Yes.
Thank you very much for a nice talk. I have a very stupid question. Because sometimes when I read the paper, then they just say, oh, I use this Mendelian randomization to show that this causal relationship. So now from your talk, should I trust it so much, or should I just say, well, you still need to verify? Because I see Nature Genetic paper, they say on upstrand, and they sound like it's a golden truth. So I just want to get your take. Should we, how much should we believe it? Or, how should we face this, or how should we do it?
Yeah. Thank you for that very nice question. So the question essentially boils down to how much do we trust each individual methods, right? Even if the methods is published in Nature Genetics, SMR is published in Nature Genetics, which is a very good journal, how much do we trust those methods?
Sorry, I didn't mean to. It's a very nice journal indeed.
Yeah. No.
Because it's such an influential statement.
Yeah.
Once you publish, it's such, everyone in the field will think it's true.
Yeah.
So I want to really get your thought on this.
Okay. So I'll give you a short version of the answer now, and thank you for giving me the opportunity to talk about my next slide. But I'll give you the answer first. So in our hands, we have realized to not trust any individual methods. So because everything when published have certain biases. So I'm sure you've seen the meme when it's published, it's like this runway model. But then, like if you really test the methods yourself, you realize after removing the makeup, it's just a regular person, right? So. And that's true. We have seen this over and over again, because when there's a publication bias, it gets into these nice journals because it shows you the cherry-picked results, right? So in our hands, I think it's best to not trust any individual methods and to test really multiple methods and see whether they agree with each other. If they do agree, that's very likely that this is a true positive. But if they don't agree, then you have to do your own tests. Which leads to my second point, which is some controversial advice for people who are starting their careers, is to make your own mistakes, right? So I'm sure you've heard of this. You've read this paper. 50% of published papers is incorrect. I'm not saying that they're all of the paper is wrong, but there's some inconsistency in the paper. Maybe a main conclusion is wrong, or maybe some figures are mislabeled. So as I mentor young scientists, I often get this question. So when they discuss with me, they say, "Oh, I've seen this result in this paper. It must be right." and that is a very dangerous mentality because it'd be right 50% of the time, statistically speaking, right? Which is almost random, right? So I think your first thoughts, I think when testing a method or reading a paper, I think the first assumption you go in is to assume this paper is wrong. And you have to be convinced that this paper is right. So to condense this is to make your own mistakes, right? You just need to test it out. And then you get this feeling yourself. And now you know whether this paper would be fitting to your study or not. Okay. Yeah.
Would you like to go on to the next few bullet points?
Oh yeah. So yeah. Full disclosure. This has worked for me. It might not work for everybody, right? So second one is put all your eggs in one basket. So by the way, the reason why I wrote this controversial advice is because these are clickbaits, right? People pay attention to clickbaits. If I write these as common sense, then no one will pay attention to these, right? So make your own mistakes. Put all your eggs in one basket. But basically, what I mean is that to focus on one thing, right? Especially for young scientists, your time is limited. Everyone only has 24 hours a day, and like, it's very hard I'd say even do 2 projects at the same time. So I say just focus on one thing and do that thing extremely well. Do that for 5 years, and then afterwards you can expand, right? And lastly, nothing beats hard work. That's common sense. Yep.
Great, thank you. Any technical scientific questions for Po Xiang? Yes.
Yeah, so just one technical question. You mentioned about QTLs and GTAGs, right? And lots of false positive. And I got this question all the time as well, so I wanted to pose this to you. And we all know that single cell data sets can be quite noisy as well, right? So I wonder if you can comment about multi-sampling and if you have calculated lot scores and things like that and incorporate that into your experimental design.
Yeah, absolutely. Thank you, Mick, for that question. So the question is that there's really a lot of noise in anything that we measure, right? And this is especially true for single cell, because for single cell, there's so much sparsity. So I don't have the golden answer for this, but what we have been doing is a few things. The first thing is really to have larger sample sizes, right? If we have a tiny sample size, then that's not going to work. So, and what we have been doing is at least sample size in the hundreds, which requires more money, right? But that goes back into the second device, is to put all your eggs in one basket. I'd rather put all my money in one project and do an excellent project rather than spreading my funding too thin to multiple projects, right? That's the first thing. And the second thing is what we have been doing on the technical side is to average the cells into what we call meta cells. So that would, hopefully average out the noises and give you the true results. Yep.
Any more technical questions? Okay, so Charlene and Woon-Tien, we're supposed to have a fireside chat, is it?
Oh, yeah. I mean,
Okay, we're supposed to have a fireside.
Okay,
You want
perfect.
to come down?
Yeah.
Yep.
So that it's a bit more casual.
Yep.
Okay, so we are having a little bit of a next stage where you can, next stage, in which you can ask Po Xiang, barring sensitive questions, anything related to work or whatsoever.
Yep.
But just to warm things up, any advice you can give for a PhD student? Imagine maybe half of the students here being PhD students at your stage. Any advice to your younger self?
Yeah. I'd say, so I'd say besides these three, yeah, so make your own mistakes and put all your eggs in one basket and work hard. I'd say seek other people's advice when you need it, right? I think that's a problem for me. Maybe that's not a problem for many of you, but that's a problem for me is I have this sort of reluctance to ask people's advice. But it's very helpful, especially if you have a mentor. To ask their advice would be very helpful. Yeah, and also I guess to proactively communicate, especially in a team setting, I think is key. Yep.
And how do you, when you first start your lab, did you sort of like, how do you manage your own small little zoo?
Yeah. How do I manage my lab? Yeah. So I don't have a very good answer for this, unfortunately. So I'm still learning the process. So I write a reflection at the year end, and I say very explicitly, these are the things that I need to try out the next year in terms of team management. But I'll say one thing that has worked very well for me. Yeah, it might not work for everyone, but worked very well for me is to do less in terms of management. So I think it worked very well for me to hire the best people and just get the hell out of their way. Let them do their thing. And it's easier for me and easier for them, right? So then the next question is that screening is very important. How do you hire the best people, right? So when I hire people, I prioritize work ethic rather than ability. Because I think ability can be learned. Everything can be learned. Nothing is hard once you learn how to do it. But it's really, really hard to change how someone thinks and acts, because that needs a brain surgery. Okay, so select for work ethic. You'd rather have someone who starts here and goes like this, rather than someone here that goes like this, right? So select for work ethic. Yep.
PhD students, you can hire them.
Yeah. So usually, I talk to them and I interview them and I ask them, like what excites them the most, and have them tell me one project that they're most proud of. And if they have really worked through and thought through every corner of that question, it's a very good correlate. It correlates very well with their work ethic. Yeah.
Is there any general advice for the PhD student in the audience? Brain surgery? No.
So most people I see, because NUS is quite selective, right? There's a lot more applicants than people who actually make it in here. So I'd say for people who are doing their research here, you're actually already past the stage gate. So I wouldn't worry about that. So I'd say most people who are spending their lunchtime listening to a talk has very good work ethic. I'd rather go for lunch rather than listening to some unknown person talking about their experience, right? So good job for being here. Yeah.
Very quiet audience. No other question for Boss Yang about how to navigate your PhD journeys? What about work-life balance?
Yes, that's a very good question. So I'm going to give you another piece of controversial advice. I don't think there's such thing as work-life balance. I really don't think so. So let me make that clear. Okay. So I think if you're working for someone in a company, then your task is assigned to you. So you plan your work days, which is 9 to 5 or 9 to 6. As a scientist, you're essentially your own boss. I say this because you're planning your own experiments and you're doing the thing that eventually will be helpful for your next step of career. You're going to be the first author or the corresponding author, correct? So you're working for yourself. When you're your own boss, you're not planning your work time. You're planning your leisure time. So what I mean, let me make that clear, right? So usually, after in a company setting, after you work 9 to 5 jobs, you do whatever you want. You usually don't do anything that's work related. You plan your soccer games. You go watch a movie. But when you're in academia and you're working for yourself, the thing is the exact reverse. You plan your leisure time, and your default mode is work. Right? So you plan to go for a movie for those 2 hours, but outside of those 2 hours, you're working all the time. So in that case, I don't think work-life balance is a well-conceived notion if you're working for yourself, right? Because the more you put into it, the more you get out from it. That does not mean, so I'm afraid sometimes people take this, what I say, in the wrong way. So let me be clear. I'm not saying that you should sacrifice your health to do work, right? You should correctly plan your leisure time. You should at least have, work out every day, work out every other day, or talk to your family. You plan these times, but outside of these times, your default mode is working. Yeah.
I would also like to add, sometimes when you're not working, especially for those PhD students who are past your QE, you are probably still, even during your leisure time, you're also still somewhat thinking about work. And that's also quite important because you need to reflect, and that is also considered partly working.
Yep.
Trying to refine in your head how to make things a bit better.
Yep.
So I think that's also quite important because if you do things like a conveyor belt, there's no time to think properly. And
Exactly.
spending thinking time, it's quite important, especially in the later part of your PhD.
Yep, I agree with that. Yeah.
Yes.
No? Yes. So I also have a question about your second controversial advice about one basket. It's really controversial, and I feel like you got to pick that basket really, really well if you want to focus only on this one.
Yeah.
So how do you approach picking a basket or a project that will lead you to presidential professorship, rising star?
Yeah, that's a very good question. So I missed something. So, okay, that's a very good question. I'm glad you asked that. I do have one more question, which is fail fast and fail often. Okay, so I learned this by working with some engineers. In the tech world, there's something called make small steps and fail extremely fast. Okay, you fail as fast as possible. So how do you do this in a non-industry setting? How do you do this in an academic setting? I say to do the most difficult experiment first. So plan your experiments. Sometimes it's not possible to know the answer, right? But let's say you have 10 experiments that you think would help you to support or refute your hypotheses, right? The default mode for most people is to start with the easy ones. So that goes. If you have the answer already, then start with the easy ones and gradually move up would be best. But let's say if you have only limited time and you're trying to put all your efforts in one influential publication for 5 years, I think it will benefit you to do the most hard experiment first. So if that experiment fails, great. Now you know that this is not worth your time, and you should move on to something else. Right? So fail fast and fail often. And once you have a good idea, then put all your energy into one project and make that project extremely well and suitable for a very high impact publication.
So I'm also going to ask you a controversial question. What if your hardest project, hardest experiment takes years to accomplish?
Yeah, so there's a few things that I could mention. So for example, for the splicing project, when we started that project, so some of my students are here, they will know that when we started that project, it's actually quite unclear whether doing splicing in single cell is possible because it's so highly biased towards the 5' or the 3' end. And there was the prevailing belief in the field is that we should not study splicing based on single cell data until we get, for example, full-length data, right? So the first experiment we did, and it took us about 3 months to demonstrate feasibility, is that we really analyzed in depth of how much is the coverage of using our single-cell short-read data. And then once we spend that first initial 3 months and we demonstrate the feasibility, then we move on to the full-scale study. So I think that was the key experiment for that paper, is to demonstrate feasibility of doing single-cell splicing using short-read data, which surprisingly people have not demonstrated before. Okay, so I think for us was the key experiment. So yeah, so I guess that was one of the examples that I can give. And the other examples is that we're always trying new things, right? So previously, we're purely a computational lab when I moved to NUS. But now, with the help of many students who have becoming more, we're more capable of doing wet lab experiments. And when we start a wet lab, right, like just the initial planning phase and buying all the equipment takes, anywhere between 6 months to a year. And the decision of going from a pure computational lab to a wet lab, that was not an easy decision to make because there's significant risk. Even if we don't do anything, we're burning $5,000 a month just to maintain the cell lines, right? So making that decision and taking that 6 months to a year to establish that wet lab paid enormous dividends in the future because now we're more capable of doing the experiments ourselves. But that initial process was very, very difficult for us. Yeah.
Okay, all right. So any last burning question? Last one. Shalene, sorry. Well, two.
Okay.
30 seconds and 30 seconds.
Okay, I just want, I'm just curious, what makes you decide to move to NUS?
Yeah. Yeah, so I think NUS is a very nice place. So there's a few reasons that I want to move here. The first one is I wanted to study Asian genetics because I did see so there was quite an influential paper in 2019. That was the last year of my PhD, which is that most of the published genetic data is European populations, more than 80%. Okay, so Asians accounts for about 10%. But Asian people accounts for 60% of the world's population, so which means that there is a gold mine to be discovered here. And in terms of the genetic diversity, as I mentioned before, I think Singapore is doing very well because of the migration history. So Singapore very naturally becomes a good place to start my career. Yeah. And of course, there's some other reasons, like family reasons. My parents and my family are in Asia, so it's natural to come back here. Yeah.
One of the top education system in the world, of course. Right?
Yeah.
Okay.
Yeah.
Last one, quick one.
Hey, Prof, very nice talk. My question to you is very often while working with the single cell data, we found that maybe one particular gene is highly expressed, and we think that gene is very important for the phenotype we are expecting. But when we knock out that gene, we don't see any changes in the phenotype, and that really means that transcriptionally that gene is highly expressed. That doesn't mean that gene is very important in driving that phenotype, right? So that leads to my second question also. How the genes are expressed is very much dependent on their chromatin organization and epigenomics, right? So do you plan to work on chromatin organization or any kind of epigenomic data in future? So yeah.
Yeah. So that's a very good question. We have not done chromatin accessibility test as of now, but I don't see why we don't do it in the future. I think it's a quite important topic. It's just we have been prioritizing one thing, which is RNA, right? And we want to really put our effort into that because there's just so much stuff that we haven't discovered. There's no reason not to move to chromatin accessibility, right? I think it will be a very interesting area to study for sure.
And how do you tackle the problem that what I stated in my first question, that we see that the gene is maybe very highly expressed, but when we are knocking out that particular gene, we are not getting what we are expecting. Then how do we choose the candidate from the single cell data?
Yeah, so I think the reason for a knockout model not performing as expected, the reason can be multifaceted. I can give you a few guesses. I don't know your specific system, but I can give you a few guesses. Are we looking at the right cell type? And I'm sure you have confirmed with qPCR that the gene has been completely, or at least 50% knocked down, right? Okay, yeah. And also, are you screening for the right phenotypes? I think we can discuss more later, because I think there's some background information to be had before answering that question. Yeah.
Okay, all right. We're slightly past the clock, but then it all leaves me to say thank you so much, Bo Xiang, for sharing your technical talk expertise as well as your words of wisdom.
Thank you very much.